Well, human biology class, welcome to Lymphatic System and Immunity, video lecture number 4, I believe. And in this one, we're going to be talking about specific immune defenses. And specific immune defenses are those which your body implements to combat a specific pathogen. It's identified and wants to combat a specific pathogen, like the coronavirus, like influenza A, like influenza B, like pneumonia. It identifies a specific strain, and it will combat it with specific resources. As we start this, we're going to talk about a couple of items that have come up before. The first of those is antigens. And on the screen here, you can see I've got an artist rendition of blood cells. That's what these are supposed to be. And if you look at the one I'm circling with the cursor here, this one has an antigen. Those are little pyramids on it. Remember, those are the markers on the surface of a cell, in this case a blood cell, but any cell in your body will have these on them that identify that cell as belonging to you, or if it's a pathogen, as not belonging to you or foreign. So in this case, this blood cell has A antigens. Here is B antigens. Now these are just the artist's rendition of the shape of them. In reality, they would be a very complex three-dimensional shape. So that'd be type B blood. Here this blood cell has both A and B antigens, both the pyramids and the circular ones. And then here's type O blood, having no antigens. Another look at this concept, because you remember blood cells have multiple different types of antigens or markers on them. In this artist's rendition, we have the blood cells, and we have green sort of triangles indicating a B antigen, or blue circles indicating an A antigen. And then there's these little black squares or rectangles here. Those are trying to represent the Rh factor. This is another entirely different type of antigen. And so here, this type of blood in the first one would be AB positive because it has both A antigens and B antigens and Rh antigens. Right below it is AB negative because it has both A and B antigens but does not have any of the Rh factor, none of the Rh antigens. So again, to remember, antigens are markers on cells that identify that cell as belonging to you or as foreign to you. Secondly is this term antibody. And this is an image from your textbook that shows an antibody. I mentioned them before when we were talking about blood. The plasma portion of blood, that clear portion of blood, which contains proteins. Some of those proteins were the globulins. We talked about alpha and beta globulins in the past, and then also gamma globulins. Gamma globulins are antibodies. That plasma clear portion of your blood contains, one of the proteins it contains is a gamma globulin, or are gamma globulins, and they have this, or are represented with this Y type of a shape. Okay, so that's to start off with. Those two terms are going to come up quite a lot, and they sound similar. So antigen, a marker on a cell; antibody, a type of plasma protein that actually is what destroys or helps destroy cells marked as foreign. Okay, when we talk about specific immunity, we're going to talk about passive forms of it or active forms of it. If you look at your notes, those are the next two lines. Passive immunity is an immunity. It's passive because your body does nothing to create it or to develop it. You are simply given this type of immunity. Specifically, you might be given an antibody. Somebody else produced it, but you are given it. An example of that would be breastfeeding, particularly colostrum milk, which is the first milk that comes into a mother after she's given birth. It is heavy with antibodies that she has developed, which actually go into the baby. And you say, well, how do they not get torn apart since they're a protein in the digestive tract of the baby? Well, babies' digestive tracts are a little bit different than adults are. Most of their digestion occurs in what are called crypt cells in the small intestine. Their stomach is not as active yet. That's why babies can't eat just anything. And so those antibodies actually do get through the gut and into the bloodstream without being torn apart. That's why that breast milk that is so important to helping a baby get through the first months and years of life. Well, months mostly, because it has not yet developed, the baby has not yet developed its own immunity. It's dependent on mom's immunities. Another example would be rabies. If somebody's bitten by a rabid dog or something like that, they would be given antibodies produced artificially outside the body that combat that rabies virus. Rabies will move along a nerve, and once it gets to the central nervous system, it's lethal. So it's important to get the rabies antibodies into a person as quickly after a bite as is possible. So those are examples of passive immunity, immunity that a person's own body does not create. Active immunity is what we're going to spend just a little bit more time on. That's when the body develops specific white blood cells to fight a specific infection that it has identified. And there's two ways that a person typically, general ways that a person typically develops this type of immunity. There's the unintentional. That's what's going on in our country right now, where there's an exposure to a virulent pathogen. That word virulent means it creates symptoms. You're constantly being exposed to pathogens. They may even infect you, but they won't always create symptoms. They won't always make you sick. We would call those non-virulent. A virulent pathogen is one that creates symptoms, makes you sick. The more virulent, the sicker you are. So you can be exposed to a virulent pathogen like the coronavirus or influenza or pneumonia, whatever. And so if you successfully fight that off, then your body will go through a process, which we'll talk about just a second, whereby that pathogen is identified. Specific resources, antibodies are developed to combat that specific pathogen and kill it off before it kills you off. Okay. Most of vaccines are really an intentional exposure of a person to a pathogen. For instance, the flu vaccine. Many times flu vaccines are actually exposing the person to a pathogen. The catch is that pathogen is dead, or it's just a part of a pathogen. See, because if the pathogen is dead, it's still going to have the antigens on its surface that identify it as foreign to your body. Even a piece of it can have the antigens which identify it as foreign to your body. So when it's injected into your body, those antigens are going to be identified, and your body will then create an antibody like this here with these specific ends. These ends of the Y here have a specific shape that kind of like enzymes need to fit that particular pathogen and destroy it or cause it to be destroyed. Okay, so that issue of intentional doesn't mean you're going to be have symptoms. It doesn't mean you're going to get sick, but it means you have been intentionally exposed to it. Okay, immunity is maintained by B and T lymphocytes. And if you remember this artist's rendition, here are lymphocytes. The artist randomly chose different colors to represent them. In a blood cell, lymphocytes are the smallest of the white blood cells, just about the same size as a red blood cell. And there are two types, B and T lymphocytes. So that's what we're going to key on next here. We're going to start with B lymphocytes and this whole antigen-antibody complex. So by a complex, I mean the antigen fits into the part of the antibody. If I go back to the picture, here is an antigen-antibody complex. Okay, this is a specific spot on the antibody, and this antigen fits into it. When that match is made, that signals the antibody to destroy the antigen that the pathogen that antigen came from. So B lymphocytes, what do they do? Okay, so from your textbook, there's a good image of what B cells, B lymphocytes do. Okay, in this portion of that image, you'll notice there are different antigens or antibodies here. They have different shaped sites. So here's one with a sort of rounded half moon shape. Here's a Y shape, and here's a square shape. So you have B lymphocytes floating around your body with many different shapes to them. And when an antigen, and here these little green dots are antigens, so these antigens have actually been removed from the pathogen. They're just showing the specific antigen. So you would have to picture these being attached to some bacteria or some virus, okay? But in this case, they're separated off. So when that match is made, like we see right here, that antigen binds to that antibody. Well, that triggers these B cells then to mass produce these antibodies. These are the antibodies here, and combat that infection. So if you look at your notes, B cells are called B cells or B lymphocytes because they mature in the bone marrow. That B for bone marrow gives them their name. They're found in blood and lymph and other body fluids. It's believed that there are more lymphocytes outside the circulatory system than in it. One researcher actually speculated that there were over 2 pounds of them in your tissue, standing guard, if you will. Okay, so the B cell, if you look at your notes, the B cells recognize a pathogen, okay, that bonding is made, and they mass produce then these antibodies here, okay, to go after and destroy whatever cell that antigen came from, and then once the infection is defeated, then the numbers of B cells decline. You don't want too many of them staying in your system because they'll find something to attack. So once the infection is defeated, their numbers decline, and all that's left is a memory B cell. And its job is to hang around with the right kind of antibody so that if that infection shows up again, your body can attack it and defeat it before you get sick. And that is the key to your defenses. Can your body identify the pathogen and defeat it before you get sick, before you suffer symptoms? T lymphocytes are a little bit different. They got a little bit of case of the specials. Okay, so this is another image from your textbook, and this is showing what happens with a T lymphocyte. You see, okay, so there's some letters here. MHC, major histocompatibility complex, type 1. You don't have to remember that. What you have to remember from this is there are types of T cells, and they're named T because they mature in the thymus gland. When I do the pig dissection, I'm going to show you the thymus gland. And they have to have that foreign antigen presented to it. There are different types of T cells. There are, what I like to use the word killer T. I think your book uses the name cytotoxic T cells. I thought killer T would be a good name for a rapper group or something, you know. Anyway, they cause targeted cells to burst by producing a chemical called perforin. Great name, because it perforates the cells. They secrete a lymphotoxin into there, and basically the lymphotoxin is a granzyme that stimulates apoptosis. Oh boy. Apoptosis is a name for programmed cell death. It's like Cinderella. When the clock strikes 12, that cell dies. There's some kind of morbid sites on the internet. There's the one called the death clock. There are actually a number of death clocks. This is a different one. And on this death clock, I put my own information in, my birthday, gender, I don't smoke, my body mass, okay, I'm a little chunky. I'm fairly optimistic. I don't drink alcohol. We're going to talk about why that's a good thing. And I'm from the U.S. So if I submit those things to this site, I'm going to find out that I'm going to die about Friday the 24th of February in 2045. I'll be 86, almost, or 86 and a half years old. There's one form of these death clocks that actually has a clock ticking, and the seconds winding down that you have left to live. So that's a little bit twisted. But that's what apoptosis is. It's programmed cell death. So this T cell has an antigen presented to it. It triggers that T cell. It activates the T cell to begin to destroy the targeted cell by boring a hole in it with perforin, inserting a granzyme, stimulates apoptosis. The cell dies. But just like with B cells, they can't be around too long. They have to have a memory T cell that would help remember that infection should it come back. There are also helper T cells that help this process along by producing cytokines, they're messenger molecules, and suppressor T cells, which turn the system off because you don't want it running when you don't have an infection to